oh my. Oh my. What is up? Beautiful morning, people. I'm awake. I was doing some some research, sitting here in bed. I figured, why not do a quick, this is light, just a light in bed little morning research stream, It has some ideas of stuff to look at recently. Anyway, so been reading a lot about fish disease, things like that, because I'm a sick man. And came across some interesting stuff. So I was trying to came across biological control stuff on protozoas. I was like, what is their natural predators? So there's actually a few interesting things. The first one I'm going to look at is from 2012. The potential utility of the leopard pleco as a biological control of the celiac protozoan Ichthyophythoris multifilus. Alright, I thought this was a fascinating title off the back. Oh my god, Ben is in the chat saying, can I have a free fish tank? This is crazy. He's awake. I mean, I'm impressed. That's all I can say. Good morning, Ben. Oh my goodness. Oh my goodness. Alright. Let's get into this. So, okay, abstract. Okay, yeah. Infections cause mortalities in freshwater aquaculture systems. Yeah, I got that. It's bad. Alright. Let's see. The free swimming protomont. What is that? Protomont. Wow. Of course, there's a 3D printer called that. Okay, protomont is the immature and pre-reproductive stage of a tomont in the life cycle of certain organisms. Okay, what's a tomont? No, not torment. Bro. Tomont is the reproductive stage of the Protozoa parasite, most notably the cause of ick. Okay, so that's the one that we see. All right. This study demonstrates for the first time the potential of G. gibiceps as a biological control to reduce multifilus on commercially valuable fish stocks in warm water systems. This is likely originated from the grazing activity of G. gibiceps on the biofilm layer upon which the multiplicative stage of the ick was shown to preferentially settle. Interesting. Therapeutic strategies against pathogenic protozoa species could greatly benefit from incorporating, where appropriate, a biological control targeting the external multiplicative life stages of the parasite. So I got onto this because I had looked at carnivorous plants, if they eat ick and other protozoa parasites and stuff like that. But the leopard pleco, I didn't expect to see that. It's a little bit bigger, so let's see what they got going on here. Got the full text. Let's see, I'm getting paywalled here. That's fine. We're gonna find it. ResearchGate. Makes it sound like a scandal when they do it like that. ResearchGate. Uh-oh. They're researching. Usually if you search the title of these papers, eventually a PDF comes up that isn't paywalled, and here we are. Okay. Let's see, is this the same one? Okay, this is a slightly different one, but this is also predation of cyclopoid copepods. So now there's the cyclops. I think that was on my list of papers to go through this morning too. So the cyclops are eating it as well. Alright. It's too early for me to be getting around paywalls like this, man. They're supposed to just give it to me, Just give me the paper, give me the info. But it's interesting that I never really read anything about where the ick wanted to settle in. So the fact that it would settle down on biofilm, maybe there is something to these cleaner fish. You know what I mean? Like, it kind of makes you think. Like, what if there's something to it after all? Let's see. Ah, there we are. Okay. Sit back and relax as we read this very digestible text that I understand. Okay, so Ichthyophthirus multifilus, so that's our favorite protozoan, is a significant global protozoan pathogen of freshwater fish in both cold and warm water environments. Infections have been reported in a wide range of ornamental and aquaculture species, including tilapia, etc., etc., etc. Okay, let's look at the materials and methods here in a second. Biological controls have been widely used for pest management in terrestrial agriculture. The removal of aquatic ectoparasites by associate species, e.g. cleaner wrasse, is well characterized in wild fish populations. See, I've been wondering, where's the wrasse equivalent for freshwater? In recent years, there have been a number of attempts to include biological controls as a wider management strategy against aquatic parasites in aquaculture activities. The utilization of wrasse and Goldstein wrasse to control infections of parasitic halogid copepods in the marine phase of Atlantic salmon has been well studied with a degree of practical success. I'm glad there's at least a degree. Other example, okay, blah, blah, blah. In spite of the strong interest, the use of biological control for pest management has remained poorly exploited to date in commercial aquaculture, where we gotta exploit this immediately. Okay, the systematic removal and disruption of the encrusted protomont stage, alright, have been shown to have been highly effective in reducing the magnitude, etc. A tropical freshwater species, Glyptophyctes gibbosips, or the leopard pleco, is a nocturnal algae / detrivorous substrate feeder that is frequently used by aquarists to maintain a clean tank environment. Is an armored catfish of the Loricariidae family, characterized by bony plates covering the body's surface. This characteristic might provide a defense against the penetration of the infected L. multifilus. Ben, you donate $100 right now, I'll give you a free fish tank. Think about it. Let's see. Okay, so that's interesting. I didn't even think about the armored aspect of the catfish being part of why they don't get certain things as much. But I can't believe that it's 6:51 a.m. and I'm timing out Ben while I'm trying to fucking read. Come on, Ben. Okay. The algae grazing behavior and the skin structure of the species. If anyone talks about my skin structure, I'm going to be creeped out. Skin structure of the species make it a strong candidate in warm water environments for the biological control by removal disruption of the multiplicative encrusted protomont stage. So the protomont, that's the reproductive stage of ick, right? And then the tomont. Let's see. No, the protomont stage is a transitional stage between the parasite feeding on the fish and its reproduction stage. Okay. And the tomont stage, that's when it's on the fish. And the trophont stage, that's when it's on the fish's gills. Oh, no, that's the one where we see it. But then there's also another cyst stage. Okay, that's when it's resistant to medication. Well, the paper's going to tell us. I don't know why I'm trying to look it up on my own. Okay. So they're thinking they disrupt the multiplicative environment on the biofilm and stuff that the ick is hanging out on, and that helps as a biological control. Okay, here we go. So materials and methods. Zebra tilapia, naturally affected with ick, were obtained from a local retailer and transferred to 32 static PVC tanks. Ben said 651. Can I have? You send me $50, I'll give you a fish tank. How about that? If you live in the U.S., and no guarantees on how big the fish tank is, but it'll hold water. I'm really just fascinated by the. Okay, here we go. So they were put into 32L static PVC tanks and temperature-controlled experimental facility within the Institute of Aquaculture, University of Stirling, UK. 20% of the water in each tank was exchanged daily. Okay. The water used for experiments was dechlorinated using a carbon filter, aerated in conditions. Okay. The fish were all fed at 2% body weight per day using floating trout pellets. All right. Solid waste removed daily by means of siphon. The parasitic infection was maintained by replacing mortalities with native blue tilapia obtained from the tropical aquarium facility at the University of UK's own reared stock. Okay, so they just keep tossing them in as they keep dying. Beautiful. Protomount settlement behavior. Okay, a natural biofilm was grown on a 20 milliliter petri dish by prolonged submersion in a 20-liter static aquarium containing water, aerated and dechlorinated water. Okay. Enriched with 15 commercial fish algal wafers and exposed to 12 hours of light and 12 hours of darkness. The dishes were periodically checked to monitor the extent of biofilm growth. On the day of use, 1/2 of each petri dish was wiped clean using paper tissue. Okay. Alright. So they wiped 1/2 with the paper tissue, keeping the biofilm layer intact on the other half of the surface before filling the disc with some water. And freshly emerged protomonts collected from the infection tank, where they are carefully dropped at the boundary between the biofilm and the biofilm-free zone on the petri dish. Then they're put through the 12-hour light cycle and checked using an Olympus SZ40 dissecting microscope. Okay, this is the microscope we gotta buy. It's only $800. Beautiful. Let's see. Every 2 hours until all the protomonts have settled, encrusted, and reached the last 8 cell stages. Then they counted the number of the protomonts on the region of the experimental substrate, and they performed it on 10 petri dishes per day and repeated over 3 consecutive days. Wow. Triplicate design with a total of 30 petri dishes used to assess the settling behavior. Okay, so this is the parasites we're dealing with. Fish stocking and rearing conditions. Okay. The use of the pleco as biological control was assessed in vivo using an experimental scale tank-based aquaculture system. 10 days prior, 6 static PVC tanks, 34 centimeters long, 19 centimeters wide, and 6 centimeters deep, were each filled with 10 liters of chlorinated water and enriched as previously described to encourage biofilm growth on the tank surfaces. Native Aureus obtained from the own weird stock were assigned to the experimental tank, and that would be the blue tilapia. They were obtained from a local commercial retailer and assigned to experimental tanks as follows. A total of 57 of the tilapia were randomly allocated to one of 6 experimental tanks, with 3 replicate control tanks, each receiving 10 tilapia, and 3 treatment tanks, each receiving 9 tilapia. Each treatment tank was then stocked with 2 of the plecos that were selected and assigned at random. The total biomass and density of the fish were balanced among the experimental tanks. With each treatment tank, the experimental design provided a ratio of 1 pleco to 4.5 tilapia, With 1,143 centimeters squared of whatever of why I say squared of tank surface. Okay, available to them for grazing. All right, so they let the fish acclimate for a period of 7 days before they started the experiment. This is they put some effort into this. I could never be this patient. During the acclimation period, the tilapia were fed a commercial floating pellet. The plecos got algal wafers to prevent them from grazing on the biofilm. Food was withheld on the day of infection. During the experiment period, the plecos were fed as described, but no algal wafers were added to the experimental tanks holding them in order to encourage them to graze on the biofilm growing on the culture system. They were put under a 12 and 12-hour light cycle with 20% daily water changes and siphoned to remove feces and uneaten food. Okay. It's getting spooky. The experimental infection was induced by adding 30 protozoans to each experimental tank when the experiment started. The infection was then left to propagate for a 6-day experimental period that allowed sufficient time for the encystment of protozoans into the reproductive stage. For the subsequent release and differentiation of tomites into theronts for the development of parasitic trophonts onto the host to a size that could be readily determined using a microscope. Okay. On day 6 post-infection, the experimental fish were killed by overdose of anesthetic 2-phenoxyethanol. Okay. And then immediately examined, dissecting under the microscope at X4 magnification to determine the total number of trophons present on the external surface of the fish and their gills. No mortalities were observed during the acclimation and experimental periods. Oh, look at them. They acclimated them safely. We love to see it. All right, so how did the statistical analysis, and I'm just curious to see how did this actually end up working. So, statistical analysis. For the in vitro experiment, a chi-square test for goodness of fit was used to determine the statistical differences between the observed distribution of encysted protozoans on the experimental substrates, biofilm versus no biofilm, and the distribution that could have been expected by chance alone. Okay. Owing to the limited number of observations, analyses were performed on each daily set of 10 petri dishes corresponding to a replicate with the triplicate experimental design. A chi-square test of independence was used to determine statistical differences in the proportion of encysted protozoans observed on the biofilm surface between replicates. For the in vivo experiment, a one-way nested analysis of variance using replicant tanks as a nested factor was applied to determine significant difference in the total biomass per tank between experimental groups and the number of trophons per tilapia between experimental groups and in the number of trophons per fish between fish species treatment group only. And now we get to the results. So did the plecos help with the population of the protozoa? Did the ick want to go to the biofilm part of the petri dish? This is what I want to know. Okay. For the in vitro experiment, a total of 275 encysted protozoans at the 8-cell plus developmental stage were recovered from a total of 300 protozoans allocated to the dishes. 95% survival rate. There's no significant differences between the 3 replicate sets of 10 petri dishes and the distribution of encysted protozoans according to the substrate quality. The proportion that settled and encysted on the biofilm surface, 50% of the total surface, was constant across the replicates, whereas an average of Sorry, okay, I'm gonna have to read it, but I don't understand it, but maybe you do. 50% total was constant across replicates, 77 to 79%. An average, where's an average? Okay. The proportion of encysted protomods observed on each type of experimental substrate was significantly different to the 50% that could have been expected by chance alone. So, 50% on each surface, because each was half, could have been expected by chance. And it looks like for the biofilm portion, 77 to 79% of the ick was on the biofilm part, if I'm reading this correctly, and then like 21%-ish was on the non-biofilm part. So they did prefer the biofilm, seems like. All right. At the end of the in vivo experiment, trophons were observed in the epithelium of the skin, fin, and gill tissue of both tilapia and the plecos. The cohabited species used in the size and ratios have appeared to tolerate each other, blah, 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 blah. In the experimental... Okay. There's no significant difference between the replicates and the mean number of trophons per tilapia in the control and the tilapia or plecos in the treatment tanks. The mean number of trophons per fish was not statistically different between the plecos in the experimental group. The mean number of trophons per tilapia was significantly lower in the treatment tanks than in the control tank, where the infection burden on the tilapia reared in cohabitation with the clown plecos was 63% lower than the tilapia reared in isolation. Okay. Morning, Jody. How's it going? Okay, so if I read that right, okay, there was a 63% lower infection burden on the ones housed with the plecos. Let's see. The study is the first to document the preferential settlement. Well, that is big. So this experiment seems to have confirmed that the trophonts have a preferred settling substrate, with 73% or whatever going to the biofilm. Jorge said, "Go to sleep, Nick. It's early." Hello, Jody. Well, why are you awake? Leave me alone, old man. Leave me alone. I'll make some scrambled eggs. The study used to... Okay, I did that. Yeah. So the first to document the preferential settlement of the L. multifilus, the ick, protomonts on biofilm substrates, and the potential of the clown pleco as a biological control agent. These findings highlight the potential of using a substrate/algae grazer targeting the external multiplicative stage of a fish pathogen to significantly reduce the parasite burden on commercially valuable fish stock. The number of trophons observed on the blue tilapia reared in cohabitation with the clown plecos was most likely due to the ingestion of the protomonts by the clown plecos during grazing on the biofilm substrate present within the rearing systems. In a previous study, a strong, significant reduction in ick infection was similarly attained using a device that mechanically removed the protomonts from the bottom of a commercial aquaculture system. The present study, however, did not investigate the stomach contents of the plecos to confirm the ingestion of the protomonts, and a simple dislodgement of them from the substrate could have interfered with the cyst normal development. Future experiments might assess what proportion of encysted protomonts are ingested or dislodged, blah, blah, blah. Hey, maybe the clown, maybe plecos and stuff, maybe all these bottom feeders really do clean the tank, man. Maybe their reputation's saved. We're back. We're back, baby. Let's go. I'm working. What's your excuse? I'm reading. It's a morning read. ICX is all I need to know. Oh my gosh. Why are we learning about ICX is all I need to know? Well, this is one particular, protozoan, but there's many other ones. So I'm on a mission like, okay, this affects this, but maybe this could apply to a few other parasites, a few other things. So we're gonna find out. I have a few papers loaded up, but this is interesting. To date, little is known about protomont settlement behavior other than they prefer to insist on light-colored substrates. Oh, great. Great. Which might provide cryptic camouflage against predators. Just getting on the sand. That's it. In this study, the plecos were susceptible to infection in spite of possessing dermal body plates. It's likely the layer of the epithelium lies over the dermal bony plates is susceptible to penetration by infective thorns. The clown pluckers were, however, less susceptible than the tilapia, and its potential as a biological control warrants further research. Alright. Alright. Let's see, anything else in here good to walk away from with this? I will put the DOI. I'll put the DOI in the chat. In the chat, if you guys are interested. But I think, let's see. In conclusion, the use of algivorous/detrivorous fish, and I'm thinking this applies to some other kinds of bottom feeders too, like isopods, shrimp. I think we're gonna be shocked at some of the things that eat this stuff, I think. But the plecos with the tilapia within an experimental tank system was found to be an effective biological control, reducing the parasite burden on the blue tilapia by 63%. This strong positive effect is hypothesized to be a consequence of the grazing activities of G. gibbosiceps on the biofilm layer that formed on the submerged tank surfaces and upon which the protomonts were shown preferentially to settle and insist. Using a biological control organism to target and remove the external multiplicative stage of an aquatic parasite has not previously been assessed and represents a significant step forward in the control of pathogenic protozoan species in an aquatic environment. Alright, and this end part is why I was interested in this particularly, even though you're like, oh, ICX, and that is, it is expected that such a biological strategy could prove to be similarly effective at controlling other parasitic diseases that have, as a part of their life cycle, a steadily multiplicative or protective life stage similar to L. multifidus. For example, Cryptocarian iterans, Amyldienium acylatum, Chilodinella piscicola, and Piscicidina. Let's look these up. What are these? Okay, what did you say? Cories? Yep, that could be cories. Okay, so the first one, that's like some other sea light type thing. Because there's a bunch of things that look like ick-like stuff for the protists and stuff. So far it looks like that's what most of these are. Okay. Now, this one I was quite looking forward to. Those saltwater fish, yeah, the saltwater fish do have like the wrasse and stuff too, so we need to find the wrasse equivalent for the fresh, the fresh water. And let me change the chat box is white, which usually isn't that much of a problem, but like it's kind of a problem right now because you can't see the chat on this white screen. So I'm like, how do I change that? Font color. There we go. Oh, this is gonna be fantastic. What color should I make the text? Should I make it pink? Should I make it yellow? When the chat comes up, that'll be readable. Oh yeah, that'll be real readable. Okay, back into this. So, this actually got me. This is the one that got me started, actually. Which is the predation of cyclopoid copepods on the thoront of ick, essentially. And I love the morning stream because I can just sit here laying in bed. Oh yeah, so lazy. Okay. So, The usual, white spot disease. It kills all the fish. It's terrible for aquaculture stuff in the beginning. Let's see when they get into. Various chemical and physical interventions have been employed. Historically, malachite green, mercurous acetate, and their derivatives were used to control due to high efficacy. However, see, listen up, Jorge. Some of us care about the long-term health of our fish, okay? What is this? However, the carcinogenic and teratogenic effects of those chemicals led to bans on their use in aquaculture. So here's the thing. I woke up late to get myself together. Oh, so you're awake. So species-only tanks are more susceptible to ick. I mean, the cleaner fish are one thing, but I think there's other stuff that could contribute to them not overwhelming. And another thing I bring up is a lot of the medications we use on our aquarium fish, aquaculture industries cannot use for fish that are going to be consumed by humans. So, for instance, here they talk about how they can't, use malachite green and this acetate because of that. On farms, formalin, copper sulfate, peracetic acid, hydrogen peroxide, sodium percarbonate, and plant extracts. Ooh, what plant abstract? I said abstracts. What plant extracts are they using? Ginger. Okay, so they're using ginger extracts and capsicum, sweet chili pepper. Okay. I honestly haven't had to deal with ick for years now. Ick has not really been my problem in general, but I'm just hoping that this kind of stuff will be applicable to other things. It's one of the more common ones, so it's a good starting point as we journey towards a world of eating parasites? ? Okay, however, repeated and prolonged treatments weaken and stress fish, increase the susceptibility to secondary bacterial infection, and may have negative environmental side effects. Oh, fantastic. Meanwhile, these treatments face challenges in open aquaculture environments, such as cage farming in rivers and reservoirs. And even though you woke up late, Jody, it's still early. There's an urgent need to discover novel, effective, and environmentally friendly methods for white spot disease. Okay. In freshwater fish farming, fish in outdoor earthen ponds with abundant plankton are not susceptible to white spot disease. But those indoor culture systems with clean water are more vulnerable. I saw something about that. Salt and temp are how most people deal with ick successfully with chemicals. But what if you want to eat the fish? Alright, so the clean water, the fish in the clean water indoors are more vulnerable. What is the potential biological mechanism behind this phenomenon? The life cycle involves three main stages: the trophont, toma, and theront. Theronts are pelagic and highly motile coelites free swimming in the water with a body size of 250 ums. Don't ask me. I think pelagic means they live in open water. They seek out their fish hosts in the water after being released from the tomots, and in aquatic ecosystems, they play an essential role in the microbial food web, utilizing the production of bacteria and phytoplankton, transferring energy and materials to larger zooplankton, such as copepods, chlorocerans, and rotifers. So they're a food source for rotifers and stuff like that, apparently. Especially the trophic link between coelites and copepods has been well documented in marine freshwater environments. The predation of larger zooplankton on coelites raises the hypothesis that free-swimming theronts could be predated upon by copepods or rotifers in aquatic systems. All right, and this looks like it's in 2023. This is relatively recent. Micrometers, Nick. Thank you for that. But kind of like more. Oh, goodness. All right. According to the author's knowledge, until now there's no information about the zooplankton predation in this report. Okay, so let's look at their methods. This is funny. Ick isolates were collected from goldfish purchased from a pet market in Wuxi, China. They just went straight to the store. They're like, let's just go buy some goldfish. The dirtiest, nastiest fish we can find. Most disease-ridden. That's where we find her ick. An indoor recirculating water system was used to maintain the ick and the fish host in a 100-liter aquarium. Underwater temperatures of 23 plus whatever Celsius, pH 7.1, all these other stats that I don't care about. All right. The juvenile gibbal Carcius auratus gibilio. I'm assuming it's just goldfish. The Prussian carp. Oh, my apologies. Weighing 30 to 50 Gs. I missed the ums. Were obtained from the experimental station of Freshwater Fisheries Research Center and used for maintaining the ick in vivo. The therons of the ick were prepared following the method described by Clayton and Price. Briefly, fish with visible white spots were immersed in aerated tap water in a 500 milliliter beaker. Oh, that sounds very uncomfortable. And left for the trophons to exit the fish and forward tomons. Tomons from the bottom of the beaker were collected and transferred to a culture dish containing distilled water. After rinsing 3 times with distilled water to remove fish mucus, they were incubated at 23.5 degrees Celsius for 18 to 20 hours. Incubating ick is crazy. To determine the concentration. Okay, what about this? 10 to ools droplets of the theron suspension was counted under a microscope. All right. Macrozooplankton collection and identification. Let's go. You can't just get this at the market. Macrozooplankton samples were collected using a plankton net 64 micrometers and divided into 2 parts. One was fixed immediately in a neutral 1% Lugol solution, and the others was transferred to the lab with pond water for the following culture predation experiments. Farm fish in the ponds were also collected and checked for infection with a microscope. The date and location of each sample, blah, blah, blah. So they got the samples from some farming ponds in China and then some wild water bodies in China. Okay. And they sampled some fish over there. So approximately 2,000 macrozooplankton individuals and 10,000 fluorescence-labeled therons were added in a plastic container with some water, containers with the same amount of macrozooplankton and culture solution but without therons served as controls. After 4 hours, co-culture under room temperature, the macrozooplankton were collected with cell strainers and observed under a fluorescent stereo microscope. To reduce the swimming and jumping of zooplankton under the microscopic view, several drops of alcohol were added to the dishes. No, bro, no. No, they did the grain alcohol and the orange juice to the zooplankton. This is crazy. Okay. For each macrozooplankton sample, the co-culture and predation trial was conducted in triplicate. 20 positive individuals from each sample were randomly selected and coded. They were morphologically identified according to the description, blah, blah, blah. All right. So let's look at this results. After 8 hours of incubation, 90% of the tomons successfully released free-swimming therons. And the therons emerged from the tomont cysts, we're going to ignore that pronunciation, presenting a 100% fluorescent signal under microscopy. Ooh, look at this. We get a little microscope photo of the ick. Look at that little, I feel like it looks cuter because it's glowing green in this. Okay, so they made them they got the fluorescent labels so they can see them. After 4 hours co-culture, no fluorescent signal was detected from the zooplankton in the control group. In the group of fluorescent labeled therons, the fluorescent signal was mainly observed in copepods and a few napoli. Oh wow, I guess like some brine shrimp type stuff in there. Or I guess fairy shrimp. However, no signal was detected in the clodocerans and rotifers and alimentary canal copepods. Several fluorescent points similar in size to fluorescent labeled therons were observed. And here we have some copepods that are glowing, which means they ate the ick. So they made the ick glow. So when things ate the ick, they could see it, is what I'm gathering. Okay. These things look disgusting, by the way. Whatever these copepods are, Zooplankton collected from 6 fish ponds and 3 wild water bodies at high speed. I wonder if this includes like seed shrimp and stuff. Okay. Co-culture and predation experiments revealed that mature copepods were the main predators of the only one copepod Napoli individual was observed with a fluorescent signal. The 28 rDNA sequences of 180 copepod individuals were amplified in the study. However, 53% were successfully obtained based on morphological characters and DNA sequence analysis. So they went all out. They look like isopods. Oh, come on. The isopods aren't that ugly. Come on. The copepod predators were identified as 7 species or operational taxonomic units of the order Cyclopodia, including, okay, you guys tell me the common name for this in the chat if you know it. Thermocyclops tyhocanensis, Mesocyclops SP1, Mesocyclops SP2, Paracyclopenia, and Macrocyclops SP. And they identified copepods, Thermocyclops tyhocanensis, and the Mesocyclops were the dominant predators. Okay, these are the ones we want, the dominant predators. No freaky, but let's see. It's not copy and pasting this right. 1 second. Thermocyclops tyhocanensis. It's interesting because I didn't know Cyclops are kind of more predators. Kind of makes me want to find some of the bigger Cyclops. So yeah, that's just the East Asia invader. Thermocyclops. Creepy little Cyclops. But it eats sick, so see, this is why people get in all those bags of copepods and seed shrimp. Oh my goodness, maybe we're all onto something. All right, let's see, what is Paracyclopena? Okay, that's just another Cyclops. These are just all Cyclops, so okay. The results indicated the predation of copepods on the theron significantly reduced the parasite burden on fish gills. And the thing is, the Cyclops are one that's actually small enough that, like, if you had a tank of bigger fish, I don't think they would necessarily hunt down every Cyclops or even really be able to, in the way, like, if you had a tank full of small predators. So, like, the interesting thing about this versus, like, a Daphnia or something is the Daphnia, like, they're like sitting ducks. They're going to die immediately. They're going to get eaten. But I could see the Cyclops, like, persisting, especially in the substrate, and since the ick settles, you know, in the biofilm regions and stuff, having a healthy microfauna seems pretty achievable and sustainable to where they are alive long enough to eat these parasites. So, all right. Let's see, how much in tap water? Looks like 84.93 trophons were detected on the first branch of the left gill in the group. However, only 54 were observed in the group with the zooplankton. Similar results are seen in the groups raised in pond water and control groups, and no ick was found. Oh, sorry, in the controlled groups, there's no ick. The water source had no influence on infection. Okay. So that's pretty cool. They died the ick and then found what ate it. Okay. Until now, few reports have been published about the predation of copepods on I. multifilus. However, most theronts with a body size of 20 to 50 micrometers fall within the prey size spectrum of macrozooplankton. In the lab experiment of the study, the authors found that the live theronts labeled with fluorescence CFDA-SE were ingested by copepods, and strong fluorescent signals were observed in the alimentary canal. Furthermore, the challenge trial shows that the presence of zooplankton in the culture system could significantly mitigate the infection of ick in goldfish. The result revealed that copepods could prey on the ick, therefore eliminating and disrupting the life cycle, blah, 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 blah. And then a little bit more over here. Planktonic ciliates are a main component of the microbial food web in both marine and freshwater ecosystems. Interesting. Because I do wonder, like, man, what is ick's role? Why does it even exist? Like, what is its role in balancing the ecosystem? And, like, in our systems, perhaps things are unbalanced. That's why you have... And things get unbalanced in nature, too, you know. Rainy season, runoff, contaminated water. But maybe if we get a bit more life in there... Which I guess, you know, the more we're planting stuff up and things like that, seems this kind of dynamic, I assume, would increase as the environment becomes more conducive to biological diversity. Look at me putting words together. All right. But their main components of microbial food web in these systems. The abundance and species composition were significantly shaped by different functional groups of the macrozooplankton predators. The cladocerans, copepods, and rotifers often co-occur in water and compete for limited food sources with different feeding and behavior efficiency. And this feeding mechanism is strongly influenced by the availability of alternative food sources. So most rotifers eat algal cells in the 4 to 17 micrometer range. Most cladocerans are efficient filter feeders, ingesting suspended particles ranging from bacteria to algae in ciliate size, so 30 micrometers. In contrast, copepods selectively feed on larger prey and consume the ciliates preferentially over alternative prey. So the copepods really like to eat these guys. In a study, 11 to 18 species of copepods, cladocerans, and rotifers were detected. However, only mature copepods were found feeding on the theronts of the ick. However, one copepod Napoli individual was also found to be positive. Okay. So one little artemia type situation did happen to feast. Oh, now they're going to the feeding strategy of the copepods. Okay, copepods can be divided into filter feeding, predatory feeding, and scraping feeding. Okay, now this is going to have some applications for me beyond this, because, you know, I've been thinking about their role in, like, fungusing on eggs and things like that. You know, I've been putting Daphne and the isopods and all sorts of stuff in my fish eggs, but I've been thinking about the role they play in that kind of thing and what they might clean up, but then also the thought obviously crosses my mind that they could decide to eat eggs, or maybe their feeding strategy damages the membrane, the surface of the egg, because much like fish, it seems like the eggs do have defenses against disease and stuff like that, which revolve around their external surface. So if any of these copepods or something happen to be damaging that, then that might be negative. But yo, what's up, Steve? Good morning. Good morning. Right now, reading about copepods eating ick and other similar things, potentially. So the copepod feeding strategy can be divided into filter feeding, predatory feeding, and scraping feeding. The predatory feeding, like, is that when you end up on one of those live streams where, like, we know you're chatting with the ick. Were you planning on eating it? Some species of copepods called mixed feeding types can filter and be predatory. I'm going to assume that, I bet that Artemia would be on that list because they look kind of scary sometimes. Food availability and body size are major factors shaping their feeding rates. Most calanoids are filter feeding. Let's look up what's a calanoid. It's just like a type of copepod, I guess. And lack the ability to hunt free swimming theronts. What the heck is this pillow talk with Nick? I'm researching. It's the morning. I don't feel like going to sit. The lights in the fish room haven't turned on yet. Not all of them. Some of them might start turning on soon. Okay. Additionally, most harpaticoids, bro. What in the name of harpaticoid? Oh my gosh! Oh, this looks gross. This looks like a scud. Looks gross. Most harpaticoids crawl around the bottom of the water body, therefore they're not detected in the water samples. Cyclopoid copepods are exclusively ambush-feeding species that hunt relatively large and mostly motile prey. I meant to look up motile. Okay, motile. A single-cell organism capable of motion. That's what it says. In this study, we discover 7 cyclopodic copepods that prey on the theronts, and we looked up some of these before. Macrocyclops albetus and Mesocyclops leucarti engage in predatory feeding on insect larvae. Ooh, how big are these things? Big Cyclops does sound scary. And this Alamy cop watermark on them is pretty hilarious. See how big is this thing? Pause. Babe, why are you looking up Macrocyclops albetus size? What's going on? Okay, varying in size by sex, with females measuring 1.7 to 4.0 millimeters and males 1.0 to 1.3 millimeters in length. All right, that's what AI says, and that's what this PDF says, and that's what the Norsuk Institute for Natural Forschung says as well. All right, they engaged. Okay, they fed on the insect larvae and such. All right, Thermocyclops tyokinensis was a fierce copepod feeding on fish eggs. See, that's why I said I was worried about. So some of them, this is a size thing. Is this copepod just bigger, and that's why it's eating the fish eggs? Doesn't look like it. Looks like it's actually smaller. Microcyclops bicolor was a mixed feeding copepod grazing on algae, protozoa, rotifers, and animal carcasses. Alright, so the cyclopoid T. tyokinensis comprised 62% of the copepods preying on theronts were detected in all healthy ponds and lakes. The absence from samples collected from ponds where fish were heavily infected with white spot disease. Okay. I mean, that kind of feels like it speaks for itself. Okay. In challenge tests, the results show the infection intensity of ick and goldfish was significantly lower in water with zooplankton. In conclusion, the present study determined that the predation of cyclopoid copepods on theronts of multifilus. The results indicate that 7 species of copepods consumed the fluorescence, blah, 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 blah. All right. That is pretty cool, and that is 2020. This is a 2023 paper, I believe. Yeah, June 2023. So this is pretty recent. Actually, I'll put the link to this in this chat. And where should we go next with this? What other diseases are parasites? Let's see. Natural predation of roundworms, fish, biological control. Alright. Biological control of parasites for fish research. Ooh, cleaner fish. Let's do freshwater fish. Ooh. Okay, solvent parasite. Control parasite. Okay, aquaculture. Okay. We got a few things loaded up here. That's not exactly. This is just control of parasitic diseases in aquaculture. Now, I want to go to biological control. Let's see, cleaner fish. So let's see. This is control of parasitic disease in aquaculture. It's parasitology, August 2022. Cleaner fish. Natural marine and freshwater ecosystems exhibit a wealth of symbiotic relationships between fish infected with ectoparasites in tropical fish farming. Mosquito fish feed on the Branchurian parasite Argulus during their free-swimming activity. What? Mosquito fish. Wow, okay, so mosquito fish eat, this is a fish louse, Argulus. Crustacean parasite. Now this thing looks gross. Apparently mosquito fish eat it during their free-swimming activity. And I thought about this before. Small fish like the Batis or the Elosoma, I have thought about if they help with parasites because they're small enough, and especially their babies are small enough where if you think about the sizes they're talking about for these parasites, at least these ones is different. They're not necessarily designed, I think, to be spread by being eaten. So if you had all these small fish preying upon them, or even small baby fish, it kind of makes sense. And if I think about it, I haven't really dealt with this kind of thing that I know of much, but maybe that's part of why I haven't really had any issues with the Elosoma or the Batis disease-wise in that sense. Because maybe they're just helping take care of the problem themselves. Smaller fish can easily recognize parasites. See, I thought about this. Okay. Small fish can easily recognize ecoparasites on often larger fish and pick them off the host skin. See, I'm telling you, I feel like this is so underexplored in freshwater because you think about some bigger fish don't really bother smaller fish. You have to wonder if there's a reason beyond the fact they just don't feel like eating them, you know? But I think that fish combinations, there's so many freshwater fish, especially aquarium fish combinations that haven't been tested. Like any kind of interaction like that happened would be hard. Because okay, this one's for wrasse, as for cleaning farmed salmon. I'm looking at the references real quick. This is a tropical cleaner fish to control. Yeah, okay, this is for Florida red tilapia. So it looks like that's probably wrasse as well. But we need something freshwater. All right, let me continue. This basic biological function is being applied by the industry by stocking salmon net pens with cleaner fish. The wrasse, yes. eating salmon lice. Okay. See, it's hard because the sea fish are being farmed a lot more. And yeah, those things did look like aliens, Jorge. A huge industry has been established in order to produce lump sucker, a species with an appetite for salmon loose attached to salmon's skin. The sustainable approach is a challenge by the high adaptability of salmon lice, formed with a lower degree of pigmentation and thereby a lower chance of being recognized and eaten by cleaner fish have appeared. This is like when you put whole shrimp in like a tank with big fish, and after like a few months, the only shrimp left are the clear ones. Okay. Various fish species predating on snails may be considered a supplement for control of digenean parasites using snails as intermediate hosts. By eliminating snails by predation, these fish may contribute to a lowered infection level. And then cleaner invertebrates. In addition to cleaner fish, removing ectoparasites from the surface of affected production fish based on predation of invertebrates for parasite control exists. The filtration of huge water masses by blue mussels can be used for trapping the pelagic larval stages of salmon lice. Cyclops predate on fish. Okay, some fish parasites here. And likewise, free-living turbellins ingest freshly delivered eggs of Pseudodactylargus, whereby the infection level for fish decreases. Which one is that? That looks like looks like a gill trematose. So is that like a gill fluke? So free-living turbellins ingest the eggs of those, and those turbellins is an unidentified species of parasitic flatworms. Wow. There's always a bigger cyst. There's always a bigger fish. There's always a bigger cyst. Okay, we got immunostimulants. I see. Caprylic acid in combination with iron and mannan, a potential immunostimulant in feed for sea bream. A Mediterranean mariculture against monogenean infections. Okay. And of course, vaccination, which I think one day in aquarium fish will be able to vaccinate our fish against certain things. I know it sounds crazy, but I think one day it'll be a thing. Okay, gut microbiota. Alright, is there anything more about... I think that's the main biological control method right now. I'm just parsing through this real quick. Yeah, I see the mussel filtration. Biological control of aquatic pest snails by the black carp. Okay. Alright, let's move on to the next one. See if we can learn anything. Okay, potential. Let's see, which one is this? I think this could be... Yep, this is a flatworm parasite. Blood flukes. Oh yeah, this is that snail one. So I think this is a fish eating the snails in order to prevent that. Let's see if we can find some more. Ooh, introduction to freshwater parasites. We might have to revisit that just for our education, a little refresher. Now I'm even smaller. The invasion of fish parasites in freshwater environments. Okay, this sounds potentially interesting. I like downloading PDFs just because then I have it. Okay, invasion of these parasites. I'm just gonna scan through for the results just because it's exactly what we're looking for, but it looks like it's got a lot of data. And look at these photos. The three most frequently recorded invasive species from left to right: the swim bladder nematode, Acrosis, the Asian tapeworm, Aschelognathie, and the anchorworm, L. cryptonyxia. Oh man, those look absolutely disgusting. Absolutely disgusting. The flatworm is also indigenous to East Asia and was first described in Lake Ogura, Japan, as Bothrocephalus acetylognathie. This parasite has a heterogeneous life cycle with only one intermediate host. Interesting. The initial dispersion of the species occurred through the importation of the species Cyprinus carpio. The common carp, responsible for everything wrong with this planet. Alright, so this is invasive parasites. That's kind of terrifying. Parasites. Ugh, this is gross. Okay. That is the snail one. I'm going to go back to my research rabbit that I had to check to see if I missed anything. Well, let's look at this University of Florida Introduction to Freshwater Fish Parasites. I just want to kind of refresh myself. Identification of a parasitic problem. Common mistake of fish culturalists is misdiagnosing and treating their fish with the wrong medication or chemical when the chemical doesn't work. They're trying another and another. Yep, I've definitely been there. Selecting the wrong treatment because of misdiagnosis is a waste of time and money and may be detrimental. Okay. Protozoa. Interesting. Most of the commonly encountered fish parasites are protozoa. With practice, these can be easy to identify and are usually amongst the easiest to control. Protozoans are single-celled organisms, many of which are free-living in the aquatic environment. Typically, no intermediate host is required for the parasite to reproduce. It has a direct life cycle, so they can build up to high numbers when fish are crowded, causing weight loss, debilitation, and mortality. 5 groups of protozoans are described in this publication: celiates, flagellates, mysozoans, microsporidians, and saucistians. Parasitic protozoans in the latter 3 groups could be difficult or impossible to control, as discussed. Oh, great. You made it sound so easy to begin. Alright. Celiates. Most of the protozoans identified by aquarists will be celiates. They have tiny, and I think this is like identified under a microscope situation. They have tiny hair-like structure called cilia that are used for locomotion and or feeding. Celiates have a direct life cycle, and I'm going to put the link to this paper in the chat. They have a direct life cycle. Many are common inhabitants to pond-reared fish. Most species do not seem to bother the host fish until numbers become excessive. In aquaria, which are usually closed systems, they should be eliminated. Uncontrolled or recurrent infestations are indicative of a husbandry problem. Many of the parasites proliferate in organic debris accumulated in the bottom of the tank or vat. Celiates are easily transmitted from tank to tank, etc., etc. If only one parasite is seen, the entire system should be treated immediately. Okay. It is an obligate parasite capable of causing massive mortality within a short time. Because the encrusted stage is resistant to chemicals, a single treatment is not efficient. Okay. Tetrahemina is a protozoan commonly found living in organic debris at the bottom of aquarium or vat. What the hell? Why would it be at the bottom of the vat? Who has a vat? I'm concerned. It's a teardrop-shaped seal. So I want to get into the microscope stuff soon, so you're going to be seeing me doing a lot of this, and hopefully one day I'll have microscope photos that we can all look at together, and some of my smarter individuals in the chat will. I love saying chat like chat. I used to stream some video games, and I always used to say it like that, and people always used to get on me like, I just can't get rid of the habit. Salient said, bro is dedicated. You've been going all night? Nope. Just woke up early. Doing a little bit of just light research for I start my day. Inspire me. Also, sometimes when I do a lot of yapping, it's like I want to make sure I get back to after a stream where I yap about whatever, complaining, I want to make sure I get back to at least one stream immediately afterwards where it's just all trying to better my brain. Okay. So it's a teardrop ciliate that moves along outside the... Oh, good morning, by the way. The presence on the body surface in low numbers is not significant. Commonly found on dead material. Associated with high organic loads. Therefore, observing them on fish which have been on the bottom of the tank does not imply the parasite is the primary cause of death. Okay. Chiladinella is a ciliated protozoan that causes infected fish to secrete. Okay, so this is something that I've been really interested in. Is that I realized some of like that white stuff on the fish, I didn't realize like the ways that fish mucus present. So I think when like trying to identify which disease it is, being able to also identify the immune response the fish is having to it could help narrow down the list. So I like that. That's what this made me think of, because excess fish mucus, like this is, I didn't realize that the excess fish mucus looks like this. And this is something that maybe I've associated in the past. Maybe I'd be like, oh, is that like some kind of bacterial infection? Which, I mean, it could be a response to that, but like the excess slime coat can be... I think this is the best photo because this is something I've actually dealt with, seen, and I was trying to figure out what it was. I think it's just useful to know, like, okay, this is like a response, you know, it gives you a hint, something potentially irritating the skin. But if it's not cysts that you're seeing, then maybe it is something else, you know what I mean? And it all has different life cycles and stuff. So the ciliated protozoan that causes infected fish to secrete excessive mucus. Infected fish may flash and show signs of irritation. Many fish die when infestation becomes moderate. I honestly think this could be part of my problem tank situation, and that's why I need to get a microscope. Many fish die when infections become moderate. 5 to 9 organisms per low power field on the microscope to heavy, greater than 10 organisms. Yeah, I'm putting this in the Discord. This would be a useful one to come back to, because it's not too like in the weeds. It's just like a simple-minded person like myself can understand this. Easily identified using a light microscope to examine scrapings of skin mucus or gill filaments. It's a large heart-shaped ciliate, 60 to 80 micrometers. Sorry, 60 to 80 m's. My apology. With bands of cilia along the long axis. Identification of tetrahemia internally is significant but untreatable problem. Damn. A common site of internal infection is the eye. Oh no. This is one of the symptoms I had of my problem tank. Significant but untreatable problem. Well, Let's just hope not. A common site of internal infection is the eye. Affected fish will have one or both eye markedly enlarged. Squash preparations made from fresh material reveal large numbers. I don't like the sound of that. Squash preparations. I feel like I know what it is, but I'm going to Google it. Squash preparations microscope. Yeah, I think it is just pressing them against the glass. They're associated with fluids in the eye. Fish should be removed from the collection and destroyed. So, as I'm looking to figure out this problem tank thing, I have now dedicated basically a fish graveyard tank because I'm not great at the destroying thing necessarily. But now I have a 5-gallon that is basically there, so when I have a sick fish that I think treatment is dubious, or I don't have the capacity really to focus on just trying to cure one fish, I can put it in that tank. And there's snails and all sorts of stuff in there, so if the worst comes to pass, then it will be taken care of. Because I'm not able to euthanize things at this point. I gotta work on that. But okay, let's write this down. We need to look this up more. Why is it so unlivable, untreatable? I go live every Saturday, 9 a.m. Like to see another early bird. I love to hit the double header stream, you know. Jesse mother effing salient. I guess I gotta stop streaming at 9 a.m. Or you gonna? I've already dedicated to not trying to worry about what other people are streaming because I'm gonna drive myself crazy. There's too many people streaming all the time, but just know that if I do clash with someone, it's not intentional. I just stream whenever I feel like streaming at this point. So I try and avoid it when I can. So, but it's just gonna happen. But I'll stop for you. Sally. I was totally gonna go for another hour or two. Trichodina is one of the most common ciliates present on the skin and gills of pond-reared fish. Low numbers are not harmful, but when fish are crowded or stressed and water quality deteriorates, the parasites multiplies rapidly and causes serious damage. Typically, heavily infested fish do not eat well and lose condition. Weakened fish become susceptible to opportunistic bacteria pathogens. Can be observed. Trichodina can be observed by scrapings on skin, mucus, fin, or on gill filaments. Its erratic darting movement, I love when they talk about its movement, and the presence of a circular tooth disc within its body easily identify it. It can be controlled with any of the treatments from Table 1. One application should be sufficient. Ooh, I like the sound of that. One application. Correction of environmental problems is necessary for complete control. That one sounds fun. It spins. Apiosoma, formerly known as Glossatella, is another... they're always changing the names. Now I'm confused. I totally knew that. Another sedentary ciliate common on pond-reared fish. Causes diseases, numbers become excessive. Organism found on gills, skin, or fins. The vase-like shape and oral cilia are characteristic. Apiosoma can be controlled with one application of the treatments from Table 1. We're going to look at Table 1. Ambiphera, previous called Scyphidia, is a sedentary ciliate found on the skin, fins, and gills of host fish. Its cylindrical shape, row of oral cilia, and middle bank of cilia identify it. It's common on pond-reared fish when present. This is why, I'm just going to say this. Besides the fact that I just don't see myself going outside in the summertime here with the weather regularly to do pond-reared fish, the extra disease factor, exposure factor is too much for me. And a lot of the intermediary hosts for fish parasites, like sometimes you'll look up a disease and be like, it's fine because the intermediary host is a bird. So your aquarium fish is never going to complete the life cycle of the parasite. Well, I got a lot of trees, I got a lot of birds near me. I live near a lot of creeks and not rivers, a lot of creeks and even the ocean. So, and even bats, there's a lot of bats. So there's just like almost certain chance of contamination. So I don't want to put anything outside that I ever want to bring inside, because I don't feel like dealing with that. I already got enough to deal with here because I buy wild caught fish all the fucking time. Although I do think the source of my problem tank, there's two main culprits. One is either a complete setup I got from a guy and it was beautiful, but there was one little white spot that he said had been on the fish and had just been there and not done anything on a cardinal tetra that I didn't see when I put it in. The other one is I got these Galaris from Jacqueline. They had the Calamala swarms, and that was the first time I saw like the cyst like thing on the side of a killifish, and I've been dealing with that ever since in tanks that fish had touched. Before that, I had almost no disease. I was new to the hobby. I had no diseases for months and months and months. It's actually really annoying because I know that my problem tank thing was definitely something I brought in. Well, of course it is. Well, actually, no, I don't know that, because it mostly affects specific kinds of fish. I only started keeping those kinds of fish eventually. Now, when I think about the problem tank thing, I'm like, I kind of think it could be something in my tap water, but I'm like, what explains the variation in its effect? As we've been reading this morning, there's a lot of things that predate, that affect. So it's totally feasible that even if it was exposed in all my tanks, it could only become a problem in a few if there was different microfauna going on. Scotty said, was this a stream just for Jody? That's what it takes. A lot of trees and birds and ew bats still doesn't stop me from tubbing. I mean, Jody, you're a much hardier person than me. You also have chickens. Okay, I had to go finish watching Stevens last night, then Nick last night to watch this one. Alright, let me know what you think. Leave a comment on last night's stream. Give me your thoughts. I was doing a lot of thinking out loud. But you better go on double time. Maybe triple time. Okay. Amphifera, previously called Kyphidea, is a sedentary ciliate. Okay. Common on pond-reared fish when present in low numbers. Is not a problem. High organic loads and deterioration. It can be controlled with an application of the treatment listed on table 1. All right, here we go. The first name in a while that we should all recognize. Epistylus. The chickens are not free-ranging. Maybe that's the problem. Epistylus is a stalked coelite that attaches to the skin or fins of the host. See, I thought of this as, for some reason, as a bacterial thing, but apparently it's a parasite. Epistylus is greater concern than many of the coelites because it is believed to secrete a proteolytic protein-eating enzyme. Maybe this is what I've been... See, all these symptoms cross over, but this is really unique. It's believed to create an enzyme that creates a wound suitable for bacterial invasion at the attachment site. Interesting. To be honest, the guppies out in the tubs look much better than the tubs in aquariums, and bigger. Yeah, I'm definitely not trying to say that tub raising is bad or anything like that. It's just it's bad for me. I like having less things like that to worry about. So for me, the negatives, and well, besides the fact that I just want to go outside anyway because it's miserable out there. Those things combined are enough. But yes, tubbing is obviously very, very successful. I was just surprised by it. This is my first year tubbing. Hey, it's your first year everything, man. Look at you. In the hobby, such a short period, you got a company, you're tubbing, you're doing it all. Living the dream. Okay, so this is... I want more photos of this wound it creates, because that could help with diagnoses, theories. Enzyme, so it's more concerned than other coelites, so other things, other like ick and stuff, because it creates an enzyme potentially It is believed that it creates an enzyme that eats protein and creates a wound suitable for invasion. OMG, Nick, the outside where it's at. I don't care if it's over 100 superhuman. I live for the sun. Well, I don't live for getting eaten by tiger mosquitoes and getting West Nile virus, which apparently was in some mosquitoes in my area. So there's some disease concerns for me personally. The mosquitoes are a real problem, which I know could be a reason to put a tub out there, but I'm gonna have to let it be out there for a while and make a difference. I'll probably put some rice fish outside eventually. We got West Nile in our parts too. Yeah, it's crazy, man. Like I said, I'm a pussy. In a contrast to other coelites discussed above, the preferred treatment for epistalis is salt. Fish can be placed in a 0.02% salt solution as an indefinite bath. Interesting. Or a 3% salt dip. More than one treatment may be required to control the problem. For more information, see the IFAS Extension Fact Sheet VM-85 Red Sore Disease in Game Fish. Okay, and Capriana. Capriana, historically called Trichophia, is a sessile coelite that attaches to the host gills with a sucker. They have a characteristic cilia attached to a amorphous-shaped body. In heavy infestations, it can cause respiratory distress in the host. One treatment from a chemical listed in Table 1 should be adequate. Okay, I don't think we've gotten to Table 1 yet. Don't laugh at me. I'm still laughing at Scotty. Said, what is this? A stream just for Jody? Well, it's 8 a.m. now. It's not even that early, so I've kind of wasted my morning talking to you guys, honestly. Several chemicals commonly used to control celiated protozoans in freshwater. I woke up at 4-ish, and I was like, I've been trying to reset my sleep schedule, so I'm just gonna stay awake. I don't know when I went to sleep exactly, but honestly, this has been a productive morning. This is good. So I'm telling you, this is knowledge is power. Several chemicals commonly used to control celiated protozoans in freshwater fish are listed below for your convenience. Okay. Most infestations respond to one chemical treatment. However, fish that do not improve should be rechecked and retreated. Copper sulfate is an excellent compound used in ponds to control external parasites. However, it is extremely toxic to fish. Its killing action is directly proportional to the concentration of copper ions, Cu++, in the water as the alkalinity of water increases. The concentration of copper ions in solution decreases. As the alkalinity of water increases, the concentration of copper ions in the solution decreases. Consequently, a therapeutic level of copper in water of high alkalinity would be lethal to fish in water of low alkalinity. There's a good note. Is something I need to deep dive in is all the different medications and how they're affected by alkalinity in reference to how likely they are to slaughter everything you're trying to save. I think Canaplex, has killed things for me multiple times now. I don't think that's necessarily related to alkalinity, but it's just, good to know that not all medications are risk-free, and also say they do have a risk, but you're generally fine if you treat them with a medication where the part of the body, let's say the liver, is what it's hard on, and your fish is suffering from something that's affecting its liver. Then that could be a recipe for a murdered fish, which I believe is something that I have done, because that is essentially me describing the Canaplex. All right. Okay, because of its agicidal activity, copper sulfate can cause oxygen depletions. Okay. Potassium permanganate is effective as well against chelates as well as fungus and external Colmenares bacteria. If we use in pond or vat, multiple treatments are not recommended as it can burn gills. Aeration should be available when potassium permanganate is used. Formalin is excellent. It is not recommended for pond use because of a strong algicide and chemically removes oxygen from the water. Vigorous aeration should always be provided when formalin is used. Okay. So, where's table 1? And then we'll come back. Bro, they put table 1 sideways. Good morning, Cam. Good morning. KJE, good morning, good morning. Let me rotate. Wow, all this nice graphics and stuff, and we have this lazy ass table. You all up early? You're up early. Jody says, morning, Cam. Chemical treatments for the control of external ciliates. X indicates the chemical should not be used for this type of treatment. What? This table looks unfinished. It doesn't make any damn sense. All right, we're going back up. Escape. Good morning, Cam says. Because there's more parasites, unfortunately. There's a lot more. I feel like we've done ick to death. We know how to kill this thing. But this is Ichthyobodo. Formerly known as Costia, a commonly encountered flagellate. Infected fish secrete copious amounts of mucus. I love the word copious. Copious amounts of mucus. Mucus secretion is so heavy that catfish farmers popularly refer to disease as blue slime disease. I'm going to read the rest of this like a true crime YouTuber. Infected angelfish also produce excessive mucus that can give dark-colored fish a gray or blue coloration along the dorsal wall. Infected fish flash and lose condition, often characterized by a thin, unthrifty appearance. Ichthyobodo can be located on the gills, skins, and the fins. However, it's difficult to identify because of its small size. The easiest way to identify is by its corkscrew swimming pattern. With a good microscope, the attached organism can be seen at 400x magnification. The organism is easily controlled using one application of the treatments listed in Table 1. Cam said, why? Okay, Crotobia is a flagellated protozoan common in cichlids. Okay, this is interesting. Anything affecting cichlids is what we're looking for, because those are the ones my mystery disease has been bothering the most. They're often mistaken for a hexamida, as they're similar in appearance. However, they're more drop-shaped with two flagella on each end. Also, they wiggle in a dart manner, whereas hexamida spirals. Crotobia typically is associated with granulomas, in which the fish walls off the parasite. See, this is what I'm talking about, immune responses. So granuloma is the fish trying to wall off the parasite. These parasites have been primarily in the stomach, but may be present in other organs. Fish afflicted with it become thin, lethargic, and develop dark skin pigmentation. A variety of treatments is being studied with limited success. All right, so we're gonna we need to look up this one more later, and the other ones they said can't be treated that well. Yeah. Okay, Piscanudinium, a sedentary flagella that attaches to the skin, fin, and the gills of the fish. The common name is gold dust or velvet disease. Okay, we know this one. Has an amber pigment visible on heavily affected fish. Affected fish will flash, go off, feed, and die. Yo, every now and then some of these descriptions are just, they're using some great words here. It's the most pathogenic to young fish. The life cycle of this fish can be completed in 10 to 14 days at 73 to 77 degrees Fahrenheit. The lower temperatures can slow the life cycle. Also, the cyst stage is highly resistant to chemical treatment. Therefore, several applications of treatment may be necessary to eliminate the parasite. Cam said you're going to need a really good microscope. I donate to the fund because I find watching microscope organisms cool. Yes, I am hoping to buy a microscope. In fact, today I'm going to be listing a bunch of stuff on eBay, even though I don't want to sell on eBay because they're so annoying, but I'm trying to raise some money for some more fish project stuff. So microscope is on the list of things I hope to buy in the next couple months. Or, well, actually really next month, because I really need to get on this. Because there's a lot of quote-unquote research I'd like to do involving a microscope. And even just things like identifying isopod species as I get into more different freshwater Acelidae. I'm going to need a microscope, and may as well go with one that is going to scale up with me as I get more into trying to understand things. So Myoxoa are parasites that are widely dispersed in native and pond-reared fish populations. Most infections create minimal problems, but heavy can become serious, blah, blah, blah. They have a spore shape and size. Spores can be observed in squash preparations of the affected area at 200 or 400x magnification or by histologic sections. Clinical signs vary, targeting the organ. Fish may have mucus production. White or yellowish nodules may appear on target organs. Chronic wasting disease is common among intestinal myozoans. Whirling disease has been a serious problem in salmonoid culture. Elimination of affected fish and disinfection of the environment is the best control. There's no remedies. There's no established remedies for fish. Spores can survive over a year, so disinfection is mandatory. Wow. See, IFAS fact sheet VM 87 sanitation practices. And the URL was not found. Okay, we're gonna have to see. This is good to know. So Jody, she's about to have 170 tanks and have a whole business and stuff. So see, Jody, she already knows all this. I should bring her on to do a talk. So I'm gonna put this link in the chat as well. Sanitation practices for aquaculture facilities. Let's see if they mention the myoxide, but no. Yep, UV light. They mentioned UV light. The ozonation. Ozonation has been used for disinfection for large marine systems for years. Ozone acts as a free radical, non-specifically oxidizes organic matter. Plays an important role in maintenance of water clarity in large display aquarium, can be helped to decrease the number of infectious particles in solution. Excess ozonation is hazardous to humans as well as fish. Ozone gas is colorless and odorless. Malfunctioning ozone generators may release ozone gas. Yeah, I have apparently my air purifier, like when I was looking up like air filters, like some of them have ozone gas leak issues. I was like, what the hell? Ozone gas that remains in solution, comes in contact with live fish, is also toxic. It damages epithelial cells and will kill the fish. I get it. It's toxic. Okay. Okay. The UV light seems a bit more reasonable. Sunlight is a practical means for disinfecting field equipment. UV light can be also incorporated in indoor filter systems to minimize the spread of infection. That's why I do think, to some degree, I feel like there was a point where it's like, independent systems of each tank is the best. Now I'm like going back, really. Actually, if you could just reliably clean all the water, a unified system would be nice. If you're going to go as extreme as like UV light and stuff, because then you don't need to do it for each tank. You know what I mean? Nick, I'm most definitely warning, but we're working on finding a lab that can test for pharmaceuticals and water samples, so we know our filtration would be enough, or we need more. Yeah, getting water tested is going to be a fun thing for me, for everyone, for you. You got the well water, and then, you know, well, you've been on the well water for a while, but depending on where you are, you know, does your water parameters change throughout the year depending on rain and XYZ and stuff like that? Now, I doubt it probably doesn't even really matter that much, but just a lot of stuff when you're just at that scale to think about. Couldn't it be me? Okay. In many cases, water from multiple tanks drain into a common sump prior to filtration or recirculation. Filtering water through UV light into the system decreases the potential for pathogenic transmission. Manufacturer recommendations should be followed, blah, blah, blah. In addition, UV light bulbs must be cleaned and replaced regularly. Ugh, just kill me. Effectiveness of UV light is dependent upon the size of exposed particles and length of exposure. For this reason, it's more effective against tiny particles. For modern recirculating systems, UV filtration is recommended. While aquaculturists must be familiar with the equipment, its limitations, and its maintenance requirements. So, not Luke Scoldies. Luckily, my water parameters don't really change. My well is 400 feet deep. Oh, nice. What's the average well depth? We did just add a filter that will remove iron, sulfur, and manganese. And I'm just going to contribute to the solution. What is average well depth in America? 142 feet, so that's what Google says. Let's see what Well Doctor LLC. Most home wells are between 1 and 500 feet deep, so 400 even by that is still at the lower end of the scale on the depthness, so pretty deep. The problem with shallow wells, so okay, there needs to be more soil to absorb toxins. Blah, blah, blah. Depth of ground. Okay, USGS. Depth of groundwater used for drinking supplies. That's not exactly how deep should you drill your well. Shallow wells, 25 to 100 deep. Moderate depth wells, 100 to 300 deep. Deep wells, 400 to 1,000 feet, or even more in arid or rocky regions. Usually drilled to reach confined aquifers and ensure water stability for a long period. So yeah, sounds good. Jody said average depth around here is 100 to 200 feet. There are 2 aquifers here. Okay, I'm just a good point. Okay. Let's finish up this disturbingly long disease list. Like, holy. Where did my disease list go? All right. Coccidia, an intracellular parasite described in a variety of wild caught and cultured fish. Their role in the disease process is poorly understood. Oh, fantastic. But there's increasing evidence there are potential pathogens. The most common species encountered in fish are intestinal infections, inflammation, and death of tissue can occur, which can affect organ function. Yeah, I bet. Other infection sites include reproductive organs, liver, spleen, and swim bladder. Clinical signs depend on target organ affected, but may include general malaise, poor reproductive capacity, and chronic weight loss. A definitive diagnosis of tissue should be completed. Several compounds have been used with success, however, consultation, blah, blah, blah. All right, this is what I really wanted to get to. Believe it or not, I'm excited. To tell you we're all now at monogenean trematodes, also called flatworms or flukes, commonly invade gills, skin, and fins of fish. They have a direct life cycle, so no intermediate host, and are site-specific. In fact, some adults remain permanently attached to a single site on the host. Freshwater fish become lethargic, swim near the surface, seek the sides of the pool or pond, and their appetite dwindles. They may be seen rubbing the bottom or sides on the holding facility where the skin or flukes are attached. They might show areas of scale loss and may ooze a pinkish fluid. The gills may be swollen and pale, and respiration rate may be increased. Fish will be less tolerant of low oxygen conditions. Gulping water at the surface may be observed. Large numbers may result in significant damage and mortality. Secondary infection by bacteria and fungus is common with monogenean damage. Gyrodactylus and Dactylogyrus are the most common genera of monogenes that infect freshwater fish. They differ in their reproductive strategies and their methods of attachment to the host fish. Gyrodactylus have no eye spots, 2 pairs of anchor hooks, and are generally found on the skin and fin of fish. They are live bearers of Viviparous, in which the adult parasite can be seen with a fully developed embryo inside the adult's reproductive tract. That is disturbing. This reproduction strategy allows for them to go quickly, blah, blah, blah. Dactylogyrus prefers to attach to gills. They have 2 to 4 eye spots. This is the other one. So this picture, we have Gyrodactylus and they do look pretty different. And they look different to everything else we've seen so far. So I do think if I had a microscope and I was able to get the stomach to collect some samples, we actually could identify some things. They're egg layers. Their eggs hatch into free-swimming larvae, so maybe this is something that can be eaten by small things. They're carried to the new host by water currents and their own insulated movement. The eggs can be resistant to chemical treatment. Ooh, excuse me. Treatment is not usually satisfactory unless the primary cause of increased fluke infestation is found and alleviated. A treatment choice is formalin administered as a short-term or prolonged bath. Fish that are sick do not tolerate formalin well, so they need to be carefully monitored. Great. That's what I'm saying. Like sometimes the treatment could just kill the fish too. Okay, then we have Digenean trematodes. They have a complex life cycle involving a series of hosts. So we get a fun diagram here. Fish can be primary or intermediate hosts. Great. Depending on the Digenean species, they are found externally or internally in any organ. For the majority of Digenean trematodes, pathogenicity to the host is limited. That sounds good. So we have the life cycle. Adult is the final form. Then we have eggs to Myricidia to Mos to Ceraceae to Metacercaceae. I'm just making it up. Then to adult. And the final host is a fish, bird, or mammal. The intermediate host can be a fish. The encysted intermediate host can be a cyst or invertebrate. All right, the life cycle most observed in fish is when it encysts the fish tissue. So this is another example of, I bet it could look like ick, but it's a totally different thing, potentially. Again, the ones that live in fish rarely cause major problems. However, in the ornamental fish industry, they're responsible for substantial digentic trematodes from the family Heterophidae have been responsible for substantial mortalities in pond-raised fish. They become encysted into gill tissue, and respiratory distress is imminent. Major problems for fish, it is readily seen and will make fish unmarketable for aesthetic reason. The best control is to break the life cycle of the parasite. Elimination of the first intermediate host. The freshwater snail is often recommended. Yeah, this is another reason I want to kill snails. They got a lot of freaking parasites. For Florida ornamental fish growers, a chemical for snail control, Balucide, is available. Interesting. Balucide is a molluscide and lampricide with active ingredient of nicosamide. It was developed to control snails, intermediate host of diseases, and is used in aquatic environment to control invasive species like sea lamprey larvae. Okay, I'm putting that in the chat too. That's something to look up later. It's kind of interesting. Alternative snail control medicine. Okay, we're getting close to the end. We're going through the worms. Nematode, also called roundworms, occur worldwide in all animals. They can affect all organs of the host, causing loss of function of the damaged area. Signs of nematodiasis include anemia, emaciation, unthriftiness. I have to look up the definition of unthriftiness because I have not seen it used this much before. Let's see, Oxford. What do you say? Unthriftiness. Lack of thriving and growth. Man, this is impossible. This site is impossible to read. Wasteful, extravagant. Okay, the quality or state of being unthrifty. Okay, I think we get the idea. Okay, Camelanus worms, easily recognized as a small thread-like worm protruding from the anus of the fish. Control of this nematode is a non-food fish. So again, just a reminder, like one reason some medications have a lot less research and stuff around them is because a lot of money in researching this stuff is for the purpose of farmed fish. So control of this nematode in non-food fish is with fenbendazole, a common anti-helminthic. Fenbendazole can be mixed with fish food using gelatin at a rate of 0.25% for treatment. It should be fed for 3 days and repeated in 3 weeks. Capillaria and levamisole works well for these as well. All the roundworms. Capillaria is a large roundworm commonly found in the gut of angelfish, often recognized by its double operculated eggs and the female worm. Heavy infestations are associated with debilitated fish, but a few worms per fish may be benign. And they also do fenbendazole for that. Okay. Here we have Eustronglides. Oh man, I don't even know. A nematode that uses fish as an intermediate host. The definitive host is a wading bird, a common visitor to ponds. The worm insists in the peronium, or muscle of the fish, and appears to cause little damage because of the size of the worm. Infected fish may appear unsuitable for retail sales. Protecting fish from wading birds and eliminating the immediate host are the best means to prevent infection. Okay, cestodes. Also, tapeworms are found. Oh my god. Look at the size of this worm compared to the fish. That is the previous one. Yikes. Okay, tapeworms found in a wide cycle of animals, including fish. The life cycle is extremely varied, with fish used as the primary or intermediate host. They affect the alimentary tract, muscle, or other internal organs. Larval cestodes, called pleurocircoids, are some of the most damaging parasites to freshwater fish. They decrease carcass value if present in muscle and impair reproduction when they infect gonadal tissue. Problems also occur when they damage vital organs such as the brain, eye, or heart. One of the most serious adult cestoids that affect fish is the Asian tapeworm. It has been introduced to the United States with grass carp and has caused serious problems with bait minnow producers. Praziquantal at 2 to 10 milligrams per liter for 1 to 3 hours in a bath is effective in treating them. At this time, no treatment can be used for food fish. Although there's no successful treatment for pleurocircoids, ponds can be disinfected to eradicate the intermediate host, the copepod. I'll have to actually start my day later on. Thanks for hanging out, Jody. Have a good day. Parasitic crustacea. Okay, we got more tapeworms. Okay, we got the fish louse that attaches to the external surface. Can be seen with the unaided eye. It's uncommon in freshwater aquarium fish. We got anchorworms, we know those. We got leeches, we know those. And yo, we made it. We made it through all the parasites in existence. This is all of them, I'm sure. The conclusion is most fish problems occur because of environment. The best cure is prevention. Good water quality management and proper fish husbandry techniques will eliminate most parasites described here. Information concerning basic fish culture, pond management, and water quality is available in the Aquaculture Extension Circulars from your Cooperative Extension Service agent. All right. That, to me, seems like a good place to end. Got through a lot of interesting concepts today, a lot of refresher on the different parasites and stuff. I feel like my brain is warmed up. I'm gonna go do some exercise, clean up, and see what I can make of the day. So, to those that stopped by live, good morning, good luck. Of course, Salient going live Saturday morning, 9 a.m. That's coming up. So feel free to stop by the stream. I'm gonna try and stay awake, get some stuff done. But it's been nice doing some reading, practicing my reading out loud. So appreciate y'all. Appreciate myself. Appreciate. It's peanut butter jelly time, peanut butter jelly time, peanut butter jelly time. Where ya, where ya, where ya, where ya. Now there you go, there you go, there you go, there you go. Peanut butter jelly. In my tanks. Out.